Hello everyone, my name is Shomik Hajra. I have done MSc in Botany and recently submitted my PhD thesis from Vishwabharati University. Regarding my teaching experience, I have taught students of class 11 and 12 for their board as well as NEET exam preparation at local coaching institute and as a home tutor for past 2 years. The topic that we are going to learn today is a very important topic from your class 12 NCERT book, chapter 5, Molecular Basis of Inheritance, and the name of the topic is the structure of a polynucleotide chain. It is a fundamental feature that you have to must learn to understand the structural features of DNA or RNA. Without this, you cannot understand the DNA RNA structure. So the things that you are going to learn today are what is ribose or deoxyribose sugar, what are nucleoside, what are nucleotide, and what is 3' to 5' phosphodiester bond. Okay, so before we start the topic, I want you to remember a trick. So what is the trick? Trick is, please send now beautiful fools. अभी मुझे beautiful flowers भेजो. Okay, so I will not remove it. We will understand how this covers the whole thing. Okay, now first let's learn about the deoxy or ribose sugar. Ribose or deoxyribose sugar. So one nucleotide is made up of 3 components. One is sugar, one is nitrogenous base, and third one is phosphate. And one nucleoside is made up of sugar and nitrogenous base. So let's decode them. Okay. So now sugars. So what type of sugars are present? 5-carbon pentose sugar in ring form. What is the ring? Furan ring. Okay, so how does it look? It is look like this. One carbon, second carbon, third carbon, fourth carbon, fifth carbon. So this is ribose sugar. The formula is C5H10O5. Now we will do 5. What is deoxyribose sugar? So here the structure is same. Here also carbon 3 structure is same. Here carbon 4 and 5 structure is same, but the difference lies in the second carbon. Difference lies in the second carbon. Here one oxygen atom, one oxygen is less at C2 position. That's why it is called deoxyribose. So this ribose sugar will be present in RNA, ribonucleic acid, and it is present in DNA, deoxyribonucleic acid. Okay, clear about ribose. Now we will go to the nitrogen base. If we move into the nitrogen base, so nitrogen base are of 2 types. One is purine and another one is pyridine. Purine are can be 2 types. One is adenine, another one is guanine. The features of purine is that we have seen the word is smaller than the pyrimidine, but structure will be larger than the pyrimidine. Let's look. It is a double ring heterocyclic 9-membered structure. Okay, So this is its structure. So why heterocyclic? Because nitrogen and carbon atom is present. So nitrogen atom is present at carbon 1, 3, 7 and 9. So let's put nitrogen atom first. Give number. So put nitrogen atom at carbon 1, put nitrogen atom at carbon 3, put nitrogen atom carbon 7, put nitrogen at carbon 9. Okay, so this is the structure of purine. Okay, double ring, heterocyclic, nitrogen and carbon atom present, 9-membered ring, adenine and guanine. And purine is can be of 3 types: cytosine, thymine and uracil. Thymine is exclusively present in DNA. Uracil is exclusively present in RNA. The features of pyrimidine is it is a single ring structure, 6-membered, also heterocyclic, nitrogen and carbon present. So where the nitrogen will be present? Let's see. So let's number the atom. Nitrogen will be present at carbon 1 and carbon 3. Okay. Now the above nitrogen base it is clear. Now we will make a nucleoside. We will make a nucleoside. So let's make a nucleoside. So first, so I have told you when OH or H according to DNA or RNA. So when we attach the nitrogenous base at the OH group of carbon at carbon 1 of OH group of carbon 1. So let's attach a nitrogenous base. So this structure is nucleoside, and the bond they will form is a glycosidic bond. The bond will be a glycosidic bond. Is complete. And if purine is the nitrogen base, then the bond will be formed between C1 of the sugar and N9 of purine. And if pyrimidine is the nitrogen base, the ring will be formed between C1 sugar and N1 of pyrimidine. Okay, so nucleoside is clear. Now we'll make a nucleotide. So let's make a nucleoside again. OH or H at carbon 2 depend on RNA or DNA. So here we know CH2OH is present. We will add a phosphate. The water will be removed, and what will they form? They will form nucleotide. So this bond is called phosphoester bond. This bond is called phosphoester bond. Ester bond is formed when acid is reacted with alcohol. Phosphoric acid reacted with CH2OH. Thus its name is ester bond, and phosphate is associated, so it is named phosphoester bond. Okay, so nucleotide. This structure is called nucleotide. It is clear. Now we move into the polynucleotide. We will join the nucleotide together. So how it will be joined? Nitrogenous base, HOH. Here always OH. don't assume here is also an nucleotide, nitrogenous base. So this OH of the 5 prime of the nucleotide and this OH of 3 prime of the nucleotide will make another bond. H2O will be removed and then they will form H2O removed and they will look like somewhat like this. OH or H. So here is it, 5 prime phosphate. So here you can see one phosphoester bond and another phosphoester bond. So this is called phosphodiester bond. Okay, so how phosphodiester bond is formed? The 3 prime OH of one nucleotide will attack the 5 prime phosphate group attached to the 5 C5 of the another nucleotide, and 3 prime to 5 prime and 3 prime to 5 prime phosphodiester bond will form. Clear? Now let's sum up it. Before we sum up, let me show you the structure. Here you can see a nucleotide, free phosphate group, and one phosphodiester bond, another phosphodiester bond, another nucleotide. Same, another phosphodiester bond, another phosphodiester bond, another nucleotide. Now these will be joined together to form a polynucleotide chain. Okay, this is structure of DNA. Huh? So from these we can denote phosphate is present in a nucleotide. Phosphate is always present in a nucleotide. From same we can say sugar, whether it is ribose for RNA or deoxyribose for DNA. From now, nitrogenous base A, T, G, C for DNA and A, U, G, C for RNA. And beautiful means backbone will be made up of sugar and phosphate. Backbone will be made up of sugar and phosphate, and nitrogenous bases projects inside like this. Look, the backbone is made up of sugar phosphate backbone is made up of like this, and the bases are projecting inside. Okay, and lastly fools say phosphodiester means 3 prime to 5 prime phosphodiester bond make the polynucleotide. So this is all for today. Thank you so much.